One of our speakers this afternoon's session, I wonder if you're in the room, that's Kees, now my Dutch accent is fabulous, as you'll guess in a minute, Kees van Bochhoff. If you are in the room, do you mind please speaking to Venkata in the front as soon as you can? If you're not, which you're not waving, therefore I'll just cry. Okay. So great to welcome the next proceeding speaker, who is going to talk to us about deep learning for Pancancer prognosis prediction, which is very exciting for me.
Good afternoon. First, I would like to thank the organizers of this session for giving us the opportunity to present our work. I'm going to talk about a model to essentially combine multimodal data for prediction of outcome of cancer patients. I'm sure I don't have to convince you that machine learning has been successful in developing models to analyze biomedical data for several heterogeneous data types. And this includes clinical data, such as demographics or family history or EHR data, molecular data, and imaging data. And many studies have shown that biomarker discovery from each of these different data types can essentially contribute to precision medicine. So what is precision medicine? Very simply put, precision medicine is the quest to optimize the treatment of patients or specific subsets of patients using biomarkers. And originally, most work has focused on using molecular biomarkers to advance precision medicine. For example, current biomarkers in use in the context of cancer patients is the Oncotype DX platform or test, for example, for breast cancer, a gene expression test to diagnose thyroid cancer, and methylation tests for colorectal cancer. And the goals of these tests are essentially to assign patients to treatments or to diagnose patients. But still, there's a lot of work to be done, and still biomarkers are needed for accurate treatment assessment. For example, triple-negative breast cancers, HPV-negative head and neck squamous carcinomas need biomarkers to guide treatments, or, for example, with glioblastoma to determine the difference between pseudoprogression and real progression. However, in many cases, molecular biomarkers might not be sufficient. And we also want to combine image data and clinical data to help decide treatment. And recent results have shown that quantitative image analysis of whole-slide images in digital pathology or radiographic images using machine learning models in radiology can actually help with this. So there's a clear need to develop models that essentially can efficiently integrate multi-scale data. So what is multi-scale data? So we essentially now have the opportunity to have data of molecules, cells, tissues, and patients. And this thanks to several different technologies. We can use sequencing, DNA and RNA sequencing that gives us data of molecules, whole slide images, immunohistochemistry, and other techniques give us image data of cells and cellular architecture, and radiographic images give us data about tissues. So, for example, on the molecular side, also we have multi-omics data. We have data from several platforms, genomics, transcriptomics, proteomics. And this is thanks to high-throughput sequencing and also, of course, single-cell sequencing, even at a multi-omic level. So there's a huge need to develop models to integrate these data, multi-omic data. Similarly, in whole-slide imaging or digital pathology, computational analysis of these images has shown that there's prognostic information in these images, and that machine learning can be used to extract information from them. But the high resolution of these images is prohibitive for manual interpretation, so that means also that machine learning can even improve upon current practice. And on top of molecular data, these images also provide orthogonal information, such as cellular heterogeneity, cellular structure, cell type composition, and so on. Lastly, also quantitative imaging of radiographic images, also known as radiomics, can provide this. And recent work in the area of radiomics has shown that you can extract quantitative features from these images or use AI models and use them to predict diagnosis, prognosis, and treatment response. And this can be done on any radiographic imaging modality, from CT, PET to MR. And essentially, again, these radiographic images provide, again, another type of information that's not available from other types of sources, from molecular data, for example. It provides data, for example, on size, location, and morphology of cancer patients. And also, it's important to emphasize that radiographic images are part of the diagnostic routine. So for many patients in the routine clinical care, we already have this data. And then another important observation is that images are more than pictures. They're also data, so we can actually use them in machine learning models. So again, to summarize, essentially, our rationale for combining this multimodal data is that each of these different types of data, molecular data, tissue-level data, and cell data, gives us information about the heterogeneity of disease in different ways. For molecular data, we actually get a lot of very detailed information about a very small piece of tissue, but not the lesion as a whole. With immunohistochemistry, we get a slice of tissue, and with radiology, we get actually the whole 3D volume of the tumor. So there's a huge opportunity to study synergy. So we developed this model using data from the Cancer Genome Atlas, and this is a unique project as it exactly gives us this type of data. We have gene and microRNA expression data, matched copy number data, methylation data, also exome sequencing data, whole slide images, and for a subset of patients, there's also radiographic images available. So this gives us a unique opportunity to develop these models. And all this data is essentially in the public domain. To give you an idea of the heterogeneity of this pan-cancer data set, there's data of about 33 cancer sites in TCGA, and these are the survival curves of the 16 least aggressive cancer sites. Here are, for example, the survival curves of the 16 most aggressive cancer sites. For example, glioblastoma is the worst survival. And you can see that each of these cancers has a very different type of clinical outcome. So this is the data that we used. We used data of about 8,000 cancer patients for 22 cancer sites from the TCGA, with breast cancer being represented by about 1,000 patients, and then chromophobe kidney cancers had 100 patients. And on average, we had about 376 patients per cancer site. This is how we set up the training for this model. We used 85% of all of each cancer combined in a pan-cancer setting for training the model. So the model was trained on a subset of each of the cancer's data, but in a pan-cancer fashion. And then we tested on each cancer separately on 50% of the data in a single cancer, but also in a pan-cancer setting. This is the data that we used. So we had clinical data, age, sex, race, and grade, which was available for all cancers. Then we used gene expression data, since gene expression signatures have been shown to be prognostic. Then we used microRNAs, because microRNAs have been shown also to be predictive of clinical outcome in cancer patients. And finally, as a fourth modality, we used whole slide images because they capture the cellular heterogeneity. One challenge that we had to take into account is the presence of missing data. And this not just missing data within modality, but also between modalities, and that certain patients did not have all four modalities. As you can see, for example, clinical data was missing in a lot of patients, but also some of these other data modalities had missing data. So essentially, we developed a deep learning network for each of these data types. The first thing that we did is we embedded each data modality in a common feature space. This feature space was a 512 feature vector, and we used similarity loss, where essentially what we're trying to do is we're trying to map each of the data modalities within a common embedding where the similarity is minimized. This similarity loss is parameterized with parameter m. When this parameter m is very big, we allow the modalities to be very different from each other. When the m is very small, then essentially all information becomes the same, and then we don't have any advantage anymore of the different data types. So essentially, setting this parameter m is a trade-off between these two extremes. And we essentially calculate the similarity loss for each patient. So that's essentially what we're going to do. So we're going to learn these similar embeddings across all these data modalities. And this has the advantage that then we are essentially, the model is robust against these missing data modalities. This is an overview of the model. So we have the 4 data sets on top. And for each data modality, we have a separate model that I will explain in detail in the next few slides. And then here in the middle is essentially the similarity loss, where each of these 512 feature vectors are being compared against each other according to this loss function. So here are the different models for each of the data modalities. For the clinical data, we use a very simple feed-forward network. Next, for the mRNA and microRNA data, we use the highway network. Neural networks are not necessarily the most optimal method for predicting outcomes from this type of data, from gene expression data. But we need a neural network because we want the model to be fully optimizable with all data modalities. This highway network was a 10-layer deep feed-forward network, and it uses LSTM-style sigmoidal gating to control the gradient flow. Next, for the whole slide images, we used a model that's called a SqueezeNet. As many of you might know, the whole slide images are too big to fit into a 2D convolutional neural network. So we also need to do a patch-based approach, where essentially we train the model on multiple patches. We essentially sample each image with 200 patches of this size, and this is, and then we combine the predictions of each patch to develop the model. And essentially, this is the SqueezeNet model. This model consists of a number of modules called FIRE modules, and then with max pool layers in between. And each FIRE module essentially has a squeeze and expand operation. And this archive paper essentially details the technical and the architecture of this model. The main advantage of this model is that it's that it uses very few parameters to develop the architecture. So this is essentially, now I have explained to each of these different models for gene expression data, microRNA data, clinical data, and whole slide images, and the similarity loss. But of course, we're not done yet, because we want to use this model to predict clinical outcome of cancer patients. And so that is essentially our second loss function, which is essentially our Cox loss that we want to optimize to predict overall survival of these patients. And we use the C-index to evaluate the model. And this is essentially a final layer that we have to the network that maps this 512 feature vector to predict survival. So this is the final model. So we have the 2 loss functions, the similarity loss and the Cox loss, that we optimize at the same time. Finally, we're also going to use multimodal dropout. So dropout is a technique that is used often in training convolutional neural networks. But we are going to add also a multimodal version of this, where essentially we're going to drop modalities at a particular probability. And then we're going to evaluate how training this model with and without multimodal dropout gives and can calculate the performance. Here are the results. So first, I'm showing you a visualization of the 512 feature vector that represents all these different data modalities. And the first striking thing is that tissue is still a very important effect when we model all this data. So I'm calling here all these patients based on their tissue. And you can see that, for example, particular tissues, for example, ovarian cancer is very separate from all the other cancers. Here is, for example, kidney chromophone cancer, and here are, for example, brain tumors with glioblastoma and low-grade glioma. This was just to get an idea, is the model picking up something useful? And we expected that tissue would be something that the model would represent. Next, we also evaluated this multimodal dropout. So essentially, we're randomly, at a particular probability, removing for each patient one of the modalities from the model while training. And what we see is that during training, using multimodal dropout consisted in a better performance compared to not using multimodal dropout. So this is essentially the yellow curve where we use multimodal dropout versus the blue curve. Here are the baseline results for each cancer, and also in a pan-cancer fashion. So this is each time the CI index for each of the 22 cancer sites, and here is the result for pan-cancer prediction of survival on the test set. Next, here are the results with multimodal dropout. And as you can see, all the values that are in green, these are essentially cancers where multimodal dropout actually improved the performance. And this is true essentially for virtually all cancer sites. Next, what we also evaluated is if pan-cancer training actually improves these models versus single cancer training. So here you can see the results. So again, for all the cancer sites, here are the CI indexes when we only train within one cancer, this multimodal model. And here is a pan-cancer version. And as you can see, only for one cancer site, we were not able to improve the performance. And I also want to point out that the improvement of the performance is very dramatic when we do this pan-cancer training. Only for the kidney renal cell carcinomas, we were not able to improve the performance. And this might be explained because this is also a cancer site that has a relatively large number of samples. Next, we also evaluated whether certain data modalities are important or not in this model. And one thing that we observed immediately was that microRNAs were the most important data modality. So again, here is the model where we integrate all 4 data modalities. And here is the model where we only have clinical and microRNA data. And for about 8 cancer sites, this is essentially the best model that we could build. Next, we also observed that we can drop gene expression data from several of the models. In many cases, gene expression data was not improving the model, as you can see here. So here, this is the model where we only use clinical data, microRNA, and image data. And in all these cases, gene expression data is not needed. But in 3 cases, the cases that are shown in red, and I apologize that this is not very visible, but in these 3 cases, gene expression data was very crucial in making the prediction. And one of them is in leukemia, where essentially gene expression signatures have been shown to be very predictive of survival. To summarize, so I've presented you a method where essentially we can use deep learning to integrate multimodal data of cancer patients and to predict prognosis. This common embedding that we are proposing essentially allows also to build these models in the presence of missing data and missing data modalities. Also, I hope I convinced you that pan-cancer learning is important and actually crucial for this model to work. And finally, microRNA seems to be one of the most essential modalities in the 4 modalities that we compared. These are the people that I have to acknowledge, but actually all this work was done by a high school student in the lab. And this is the funding that I have to acknowledge, NIH funding from NIBIB and NCI, and also from the Innovation in Cancer Informatics and from the Parker Institute for Immunotherapy. And all code is available from GitHub. And thank you for your attention. I'm happy to answer any questions.
So I have 2 questions for you. One is, I'm curious about how you handled the clinical data, because the fact that patients got different lines of treatment may have something to do with how long they lived.
Yes.
The second question for you is, how do I get one of those high school students?
I definitely wanted to emphasize that this was done by a high school student. So, and she will be going to MIT next year. So the clinical data, yes, of course, these patients are very heterogeneously treated, and we cannot control for that, right? So we're trying to predict overall survival, and overall survival is a very distant outcome of these patients. So we would more likely to predict treatment response, but in that case, we probably wouldn't be able to do it in pan-cancer fashion, because then it would be more specific to each cancer site.
I guess the nice thing about this is that be it TCGA, they're all pre-treatment, mostly, almost all of them are pre-treatment diagnosis level.
Yes, for sure. All the data is so the molecular data is pre-treatment, yes, and also the images.
Hi, thank you for the talk. I have a question regarding, I fully agree, and I strongly believe that we need to combine all these different modalities in order to better understand generally the tumor biology and the mechanisms. However, when you think of a clinical application as a prognostic test or a companion diagnostic or something, I think you would certainly prefer a more simple test, because you don't want to run multiple different assays before you can have a result. So can you comment on that a bit?
So I think that's what I try to show you, and is to a certain extent with the essential data modalities. So this is not something that we can automatically derive from this platform, namely which modalities are crucial. But for sure, yes, I completely agree that we want the most simple biomarker diagnostic that is also the most cost efficient. So that's definitely part of our future work, is to determine which patient needs which data modalities, and can we start with the modalities that are clinically available, such as clinical data, radiographic data, pathology data, and then molecular data for the complex cases.
So in the spirit of honesty, I'll tell you we have a slight problem in that one of our speakers for this session has a delayed flight. The good news is that means that we can give this more question time, because I can see how many hands are up in the room. The bad problem is that we've got to figure it out later. Venkata. Right, I think there's one here, one there, and then, no, you first, and then you. Okay.
So, nice talk. You showed that cancer type is associated with survival, and so I was just wondering if you tried to correct for that at all, like, or maybe tried to predict survival based on cancer type alone and see how that compares with using these different types of data.
So there was only one line in the results where we actually look at the prediction of survival across all cancers. And in that model, yes, I agree, we could take in the tissue and see how well the tissue on its own already predicts survival. And very likely it will predict survival to a great extent, right? Because that's essentially the plots I showed you in the beginning. But when we are testing within each single cancer site, then that becomes irrelevant, right?
All right, 2 more questions. You shouldn't give such a good talk, Nick.
Hi, thanks for the talk. One question, would the model even improve, or could the model be improved when you would give him an idea which microRNA regulates which mRNA? So basically, if you could, for example, code that as a graph and then have an embedding from a graph neural network and give the information as well?
That is definitely a possibility, but it might also make the model much more complex, right? So, and yeah, that we have done something similar using more a kernel framework, where we essentially combine not imaging but multi-omics data, and we use essentially an additional kernel, which is a graph that then links with a similarity matrix multiple omics data sets. And we could show that that's could also, in the same sort of setup where we had multiple data sets to predict overall survival, where we could improve the models. I would not immediately know how to translate that into sort of like a deep learning setting. Yeah.
Thanks.
Right, last question.
So for the TCGA cohorts, for many of these, for instance, breast, we have with rather trivial methods like Cox regression, we get CNC index of 0.7, maybe even until 0.98, which is maybe 10 percentage points different from your method, but using much less data. For instance, for some of these signatures which are used, these are just gene expression signatures of 20 or so genes, right? So the first question would be, do you see your method as a significant improvement compared to this kind of very simple methods? And second question is, would it be possible using your method to go back to the original features and look at the feature importances of the original input features in the spirit of interpretability, and see whether you come up with different or similar features as being important, which then also could be tested in the clinical setting?
So to the second part, yes, for sure, that's definitely something that we can look at, is which features are important in each single modality and then across all modalities. For the gene expression microRNA, that's relatively straightforward to do that by backpropagation. And then for the images, we could look at which patches are the most informative, together with all the other types of data. And then for the first part of your question, I think it's not straightforward to compare this with more traditional machine learning methods because there's not many methods that can also look at the image data. So it's not clear how we would be able to develop such a model like a logistic regression or something like that.
Okay. I'm sure lots of us are going to corner you over coffee. Thank you so much again for a wonderful talk. Okay.